is Peter Daddona from Zosano. And I know the video guy's getting ready, so I'm just waiting for a second here until they get your slides, Pete. So our next speaker is Peter Daddona from Zosano, formerly Alza, and a friend and colleague of some of ours. So Peter, I'll let you take it away from here.
Okay, thanks very much, Chris. And I thank the organizers for inviting me. I'm not a chemist. I don't know how to make peptides, but I've been working with them from the biology side, from the engineering side, from the product development and formulation side. So, there's the whole spectrum. And what I'm going to focus on, and Frank did a very nice job kind of going through the whole history of classic transdermals and the innovations to get hydrophilic molecules able to cross the skin barrier for transdermal, opening up really a lot more possibilities. And, as the questions came up, it depends on, okay, what's the system? And what's the system depends on what you're trying to do, what drug you're trying to deliver, what kind of PK profile you want to achieve. And this kind of, if you work backwards, you're saying, what's the target product profile? What do I want to do with a delivery system? And then, can I do it with this system? What are the technical challenges, and how do I go about solving them? And so I'll give you a little bit of the journey that we've taken with the microneedle technology. We use solid microneedles that are drug-coated. So we believe we're the most advanced because we're the closest right now to a commercial product in terms of manufacturing, in terms of clinical studies, in terms of regulatory discussions, et cetera. We were formerly known as the Macroflux technology. We came out of Alza. Alza was bought by Johnson & Johnson, and then we ultimately spun out the company because there were not enough products in the J&J pipeline for us to work on. There weren't enough things of significant interest for a big company like J&J. But we liked that opportunity. We raised venture capital money. We're still venture backed. We selected very early in our process parathyroid hormone 1 to 34. And that's currently the only anabolic peptide which is approved for treatment of severe osteoporosis. And we're just about ready to enter phase 3 with our clinical trials. So why did we pick parathyroid hormone 1 to 34? Well, first of all, parathyroid hormone 1 to 84 is a naturally occurring peptide. If you have an overproduction of PTH with hyperparathyroidism, you'll get bone loss. If you give PTH on an intermittent schedule, once a day with a short burst, you'll get bone formation. So this is another one of these cases where peptide hormones are very, they have to be given in a particular way to get a particular effect, or you may get the wrong effect. So you can't just say, I'm going to give something once a week or once a month and expect it to work biologically the way you expect. In some cases, you can get away with it. In other cases, you'll have a very suboptimal product. As I said, the product that's on the market now is Forteo. It's a sub-Q injection of PTH 1 to 34. It has to be injected once daily, and the duration of treatment is about 2 years. And again, the sub-Q profile is sufficient for approval because it was demonstrated in clinical studies to prevent fractures, bone fractures. So when we looked at the product opportunity and looked at a transdermal alternative to the sub-Q injection, we wanted not only the convenience, compliance, room temperature, stability, but we wanted to have a very rapid PK profile in order to drive the anabolic reaction and to get an equivalent or even better bone strengthening and reduction of fractures. So this is what our technology looks like. It's the patch itself is about the size of a quarter, adhesive backing, and in the center is the metal, we use titanium or we use stainless steel, but it's a microneedle array. If we take a small section of that and blow it up and we magnify it, you can see that the design here is like little arrowheads. For a 2 square centimeter array, which is shown here, we have 1,300. And more importantly, we can fabricate these, but we can coat these with drug such that the coating is right on the tip of this microprojection. And the reason we do that, the reason we localize it, is because when these microneedles go into the skin, we want the drug to go into the skin. So this is, if you will, like a direct injection, where we're administering the solid drug coating into the intraepidermal or intraepidermal space, where it can pick up the interstitial fluid. That coating can dissolve. The drug can then diffuse from a very high local point concentration and go where we want it to, which is basically to the capillary beds and to get a rapid where we can get a rapid absorption in the capillary bed. So here it's shown when you apply the patch to the top of the skin, the microneedles that have the drug coating go into this intraepidermal space. They then dissolve and then the coating will diffuse. So this is a totally passive system whereby we just have to apply the patch onto the skin. And when we looked at this very early in the development cycle, we said, how are we going to do this so people would know how to put the patch on the same way each time? If they put it on a different site or a different person decides to put it on a different way. Well, it's very hard to train anybody to be able to do something consistently. That's just human nature. They'll find another way to do it, and it probably won't be right. So we developed an applicator which takes the control of the patch application and allows us to do this reproducibly. This applicator is handheld, it's reusable. We also have one which is a single-use disposable, which is very cheap. But the reusable one is very robust. We can use it over 1,000 times and not get any fatigue. It does the same thing each time. What does it do? Well, we take the patch and we put it into a ring so it holds the patch, and then it docks to the applicator. And it just docks by a pressure. You just click it, the ring goes on, the system is ready, and then you press and it automatically applies the patch. So what happens during that process? Well, during that process, there is a hold down force which allows the skin to stretch. This is very important because skin is viscoelastic, so you want that skin to be taut. And so person to person, site to site, you can control that skin tensioning. That's the target for the microneedles, so they can penetrate into the skin. The second is, once you achieve that sufficient hold down force, it trips a lever and there's an impact spring which drives a piston, breaks the patch, and applies that microneedle patch onto the skin with the right time and force. And we worked out the conditions for this such that for the system that we have, we know that we can get reproducible microneedle penetration. So everything that we need to control the patch application is built into the applicator itself. As soon as the patient hears the click that the patch has been applied, everything's been done the way we would like to have it done. And again, it's a simple press and apply. Patch wear time for these systems, because they're drug coated, in this version we're not using a reservoir, we're just tip coating these microneedles with the drug. And we're using very hydrophilic excipients, and the drugs themselves are very hydrophilic. We get very fast dissolution. We get very efficient delivery off of the patch and into the skin. So I put down here a 30-minute wear time, but in reality, all of the drug is off the patch in the first 10 minutes. And the advantage of that is that if you forget to take the patch off after 30 minutes, there's no more drug coming out of the patch. There's no risk of overdose. So that's an important safety feature. If you fail to do that, there's no safety issue. Okay. The next was we have a system that we like. Most of the people who are scientists and chemists and process engineers. I mean, we do hallway marketing, but we took this outside and we said, okay, let's look at our target patient population, which is postmenopausal women with osteoporosis. And we did this study in New York, in Dallas, in San Diego, and in San Francisco, 288 postmenopausal women. We gave them picture directions on here's how you should apply the patch, and gave them the system. And we were very pleased that the ease of use was high, the concept was good, but most importantly, it was intuitive, that just seeing the pictures on how to do it, 82% did it the first time, 100% got it after one intervention by the facilitator. So this is key because if a technology is too foreign, people won't use it. I think there are some examples of some failed products which were just too complicated. It looked interesting. The patients tried it once and then they said, I don't think it worked, or I don't like it, and then they don't use it again. So having good usability and this kind of feedback is very important to developing a successful product. Okay. All right. Now a little bit about the product itself. So when we look at drug-coated microneedles, the first question we ask is, can you open the package and get at it? The second is the product stable in that package? And because we're dealing with a peptide hormone, stability is a key feature. The marketed product is a liquid formulation, and it should be kept refrigerated. We are developing a dry formulation, and we would like to get an extended shelf life stability. We do that in a couple ways, but the most, or I should say the easiest one for me to explain is the packaging itself, where we do a nitrogen purge to exclude oxygen, and we have a desiccant present, which basically takes our relative humidity down to less than 1%. So we have less than 1% oxygen, less than 1% humidity. The formulation itself, once it's dry, is very stable, and I'll show you some data for that. And the packaging itself is in a vapor impermeable foil that's sealed. So you just open it, you dock your applicator, and then you press and apply. Okay. So here are the stability data, and we compare this to the marketed product, which is Forteo. And here is shown the stability curve for the refrigerated liquid formulation, which is not so bad, but because here we're looking at, I can't really see the numbers, about 85% if you extrapolate out to 2 years. And if we look at our dry formulation, this is real-time stability data at room temperature, and we're only getting about 1 or 2% loss in molecular integrity. No, of course, no loss in potency because those assays aren't very tight. But the molecular integrity for this small peptide is excellent. Okay, so there aren't any engineers, or maybe there are wannabe engineers in the room here, so I'm just going to quickly kind of go through the system, because if we talk about making a drug delivery device or system, you got to be able to make it. And we like to keep things simple because they get complicated on their own. So I'm going to just briefly describe a 3-step process that we use to create this type of patch system. Obviously, the formulation is very important, and we generally work with high concentration peptide formulations with stabilizing excipients. We have to coat these onto the microneedles, and basically we start by making the microneedles, and we use a photo tool which has a design. We do an etching step and a forming step, and this creates the microneedle array. We then have a system whereby we put the rings together with the patch and the array, and we call this a ready-to-coat patch. So once we make this, they're like poker chips in a Pringles can, and you put them on the shelf. So you can coat them with PTH, or you can coat them with drug X, Y, and Z. They're just ready to coat. And the second, I should say the last is, okay, the coating machine itself. How do we put the coating onto the tips of these microneedles? and how do we package it. And we have a scalable process that we use to do that. Okay, so the first is the etching process. Because of the nature of the way we set up an AutoCAD and the photo mask, we can create any design we want. We can etch it. And here's an example of some of the different shapes that we can get. And they all have different geometries, so they have different loading capacities for the drug. The key features, though, are really the tip angle, because that's what has to penetrate into the skin. And the other is just how we can coat onto these geometries to get it to be very uniform. And this etching process can go reel to reel, which will give us very high throughput as we move down the road. The next is the equipment that we use, which creates these ready-to-coat patches and puts them in a holder. And here's the business end, which basically inside this machine there are reservoirs, and these reservoirs contain the formulation of the drug. So if this is PTH, we have our high concentration, high viscosity formulation. We have a drum which will roll, and this particular one says it's at 50 RPM. And what that does is it allows the formulation to get picked up by the drum and to form a uniform liquid film. Then we're able to pass that patch into that liquid film, and by doing so, it's a dip coat. It picks up the liquid formulation and it dries. And you can repeat this process several times to pick up the coat weight. So you can see here's 1 coat, 6 coat, 8 coat. So you can control the process either by the concentration of what you load into the reservoir or the number of dips that you do in order to create the ultimate drug load that you want on your microneedle array. The other way to change the dose is just by the area of the patch. And you can go from within this template, which is a 5 square centimeter template, you can go from 1 to 3 and even 4 square centimeters. And so you can get several doses, not infinite numbers, but several doses. And this is what's used in standard transdermals. You just change the area of the patch to change the dose. Everything else in the process is the same. Formulation doesn't change. The coating per micro needle doesn't change, just the area. Okay, so where are we? Right now we're right here. We're making clinical supplies with this equipment. We have a Japanese partner who is committed to working with us to make the commercial manufacturing. The commercial manufacturing will be about 8 to 12 million units per year per piece of equipment, and you can add 3, 4, 5 lines in order to build your annual capacity for production. Okay, so that's kind of the process development, that's the equipment, that's the way we scale that to get to commercial volumes. Now I'll go back to the drugs themselves and what goes on the microneedles. What have we done so far? And then I'll focus really on PTH. We've done 12 clinical studies, over 400 subjects, and 30,000 patch applications. So I think a question was raised about tolerability, red marks. Is this something that patients or subjects are going to like? We have a lot of experience now that says that isn't a problem. We've also done a number of preclinical studies, and really the gamut there is to look at the boundary conditions for the technology. What can it do? What can't it do? But of course, you have to focus on a couple things that you are going to move forward, because there's so much involved to move any one product forward through all of the clinical and regulatory and product development hurdles that there are. Okay, so this is the universe of things that we've looked at, which go from small molecules up to monoclonal antibodies. I would say that we've delivered 1 milligram of monoclonal, which doesn't really fall into a therapeutic range for anything that's of current interest. We've done a vaccine and got successful phase 1 results with a flu vaccine, but I'm going to focus on the PTH and talk particularly about clinical performance. So the first question we asked when we put PTH on our microneedle patch was, okay, what does the PK profile look like, and what is the site of application, what effect does that have on the PK profile? So this is a little bit complicated in the sense that a couple things going on here. One is that these are normal volunteers, and first we did a sub-Q injection of the marketed product, which is Forteo. The Tmax is about 30 minutes, and you can see what the AUC curve looks like. This was a thigh injection. We've also done an abdomen injection. And then we put the patch on 3 different sites. We put it on the abdomen, we put it on the upper outer arm, and we put it on the thigh. Because this was a phase 1 study, we remove the patch. We measure how much residual drug is left on the patch by extraction and HPLC. We do a skin swab, and we ask how much is on the skin that didn't get absorbed into the skin. And then by calculation, we say, okay, X amount actually got into the skin. And in general, it's about 80 to 85%. And that's what we saw when we put it on the arm, the abdomen, and on the thigh. The delivery into the skin was equal. However, when we looked at PK, we saw that absorption was best at the abdomen, slightly lower at the arm, but significantly lower on the thigh. And this is probably due to some absorption differences that we have not pursued, because our really preferred was going to be the abdomen as a site where we wanted this patch to be delivered. But it's interesting that you do have to look at site of application, and clearly, even if you do injectables and it's sub-Q, that's going to vary with fat content. That's going to vary patient to patient or subject to subject. So you do have to prescribe where you're going to make that injection. The next thing we looked at was patch wear time, and we wanted to ensure that all of the drug was coming out within 30 minutes, and that if we left the patch on longer, we weren't getting more dose coming out. And that's just what this curve showed, was that either leaving the patch on for half an hour or leaving it on for 2 hours didn't make any difference. And subsequently, we've done 24 hours just to satisfy some safety concerns about, well, what if the patient didn't take off the patch? Is there going to be more drug coming off, or is there going to be a tolerability issue? And the answer to both of those was no, it didn't make any difference, because the patch itself is a very superficial interaction with the skin. Okay, so now I'm just going to take you through a phase 2 clinical study, and this is not only PK, but it's looking at PD. It's looking at what, are we seeing the drug delivered with the right PK profile, and what is the marker for efficacy? We did this as an outpatient study where we train people at the clinical site. Here's how you put the patch on, give them a 30-day supply, send them home, check on them with a phone call to make sure they're not having any problems, and then a month later they come back, they give us their used patches, they tell us any experiences that they had, and then they continue. This was a 6-month study, outpatient. This was done in 3 countries. It was done in Argentina, in Mexico, and the United States. Had 165 patients enrolled. We looked at several doses of the patch, 20, 30, and 40 against the Forteo, which is a 20 microgram fixed sub-Q injection, and a placebo control. The primary endpoint in these types of studies is not fracture reduction, because those studies take years to do. All of the regulatory agencies have approved that looking at an increase in bone mineral density, which you do by a DEXA scan of the vertebral spine, is sufficient to look at the increase in that bone mineral density over time. For a product approval, it's 1 year plus a 6-month safety. For this phase 2 study, we just did 6 months because we wanted an early read. So we did that for vertebral spine, and then as a secondary endpoint, we looked at hip, because hip is a very important bone in terms of fractures and morbidities and mortality effects and cost effects for healthcare. And of course, we looked at safety. And with these types of agents, the first thing that people worry about is hypercalcemia. if you're getting too long an exposure to PTH, and of course topical effects. So the first thing I want to share with you is the PK data where we took samples at 1 month and 6 months and we compared the PK. The first in red again is the Forteo product, which is a sub-Q injection. And then we look at the 3 doses of the patch. And it's really hard to see, but there is a dose proportionality in terms of the amount delivered. If we look at AUC, and this is important, this is looking at inter-subject variability, but the data are the same intra-subject. So the coefficient of variation was the same. What's striking is that in this study, if you just look at a sub-Q injection and you look at the intra-patient variability, it's remarkably high. So here's the mean, but we have some pretty big extremes. And here are the 3 doses of our patch. We're a little bit lower, about 80% lower with our highest dose than what we see with the Forteo, 20 micrograms. But the CV is really no different when we deliver by the patch or we deliver by the sub-Q injection. And again, remember, this is an outpatient study where subjects are self-administering over a course of 6 months. In this case, the bioavailability from the patch was 40% overall. That's based on the coated amount and the plasma concentration relative to the Forteo injection, which is about 95%. So the first set of data were the primary endpoint, which was vertebral spine and looking at the increase in bone mineral density. And again, you do a DEXA scan, which is a routine scan that all postmenopausal women are getting once a year or maybe once every 3 years to look at whether their bones are getting less dense and maybe they're at higher risk for fracture. So this is a standard procedure. And so in this clinical study, we looked at patients who were not on drug for 6 months, and you can see there was a slight decrease in their bone mineral density at the vertebral spine. But as we put the patch on and we look at our 6-month time point, we do see a nice increase in that endpoint. And again, this is measured against another arm, which was just Forteo administration. So if you just eyeballed it, you would say that, okay, our 30 patch looks the same as the Forteo 20, but the study really was not powered to look at that difference. It was powered to look at the difference between patch, active treatment, and placebo. But we think we're okay right around this area in terms of getting something that's going to have a similar or maybe a better effect in the vertebral spine. Then we looked at our secondary endpoint, and this was a little bit more striking. Again, you see a drop in placebo bone mineral density, which again is expected for this bone. Forteo, there's not much going on at the hip, and this is known. At 6 months, you don't really see much of an effect with Forteo, but at 1 year, you start to see it. So we interpret our data to say that at 6 months, we appear to be getting an earlier and a significant effect in the hip that we don't see with the Forteo. At least in this study, that's what it suggests. And, question is why? Why would that be? And we have key opinion leaders who are espousing certain mechanisms of driving anabolic reactions and lowering bone turnover because of the pulsatile PK profile, which is even sharper than Forteo, and this could be driving the effect. I think longer and bigger studies will confirm what we're finding in this study. So again, the outcome was that we were getting a similar effect at the lumbar spine. It appeared we were getting a more early and maybe an improved effect at the hip. We did not see any adverse events which were different than the marketed product. The topical effects were minimal, and they were just a mild erythema, which would resolve in less than 24 hours. And it's a pink spot, not a red spot. There were no skin infections. People worry about, well, I break the skin, and what about getting adventitious skin infection? Again, we've been using this technology for a number of years. We've never seen it. What we do is that we have a sterile product, and at the point of use, we ask people to clean their skin. It doesn't sterilize it, but clean it with alcohol. just removes the dirt, the oil, and makes one person a little bit more similar to another. And then you can apply the patch. And I think really the advantage of the alcohol is so the adhesive sticks because it's a patch. So we saw no skin infections, even with people who had high staph counts on their skin. And most importantly, we saw no evidence of an antibody response, because people always worry about, okay, you're putting something into the body. Is it going to be recognized as self? Is it going to be something which is, you're going to make an antibody against it? And we saw no evidence of any antibody formation after 6 months daily treatment. So basically, we're doing prime, boost, and challenge all in this study, and we're seeing no evidence of an antibody response. So we think that this validates our technology platform. Like in most things, so far so good. What's the path ahead? We do have our Japanese partner who's committed to getting this product commercialized, first in Japan, but they have rights for Asia, China, and Taiwan. They're committed to working with us to set up commercial manufacturing in Nagoya, Japan. They have a vested interest in the osteoporosis field because they'll be marketing Reclast, which is a once-yearly IV. They have a salmon calcitonin injectable, and they have just gotten approved their own Japanese PTH for a once-weekly, and so they're very motivated to have a patch to come into that Japanese market with us. I'll just make a comment about the journey of getting buy-in from the regulatory agencies about what you're doing, how you're doing it. It does take a number of interactions, particularly with a new technology. There's an education process. They all claim to be good scientists at heart and bureaucratics by necessity. So you have to prove to them that it's reasonable. They have to get a certain level of trust. And then, of course, you've got to check the box and do all the things they ask you to do. If it's too onerous, then you have to negotiate. But, that's the name of the game. We have gone to the U.S. agencies, Japanese agencies, and we've gotten scientific advice from 3 European countries. And so we're very confident that this technology will pass the regulatory hurdles, provided our pivotal clinical data comes up to snuff. So I think I will kind of leave it at that, except just to say that for anybody that's embarking not only on formulations or new chemical entities of peptides, or using a device to aid in the delivery, the bottom line is that you need staying power in this business. If this is not for especially in the pharmaceutical business, this is not for the faint of heart. You've got to stay at it. Persistence is a huge factor. Capital backing is a huge factor. Having a good management team. You have to believe in what you're doing, because if not, you're guaranteed the outcome will be failure. Even when we think we've got a very good system, the risks are still in front of us, but we're confident that we're taking all the precautions that we can get there and we can be the first to market. So thank you very much for your attention, and I'd be happy to answer any questions.
Hi. Are your patches and microneedles silane-based?
Am I what?
Are your patches and the microneedles silane-based polymers?
Silane?
Silane-based? Are they silicone?
Silane? No. So we've gone, well, there's different kinds of microneedles. We've gone with metal microneedles.
Okay.
So we etch the metal. It can either be stainless steel or it can be titanium. There's some different advantages to one over the other. They've been silicon-based microneedles, as we've talked earlier, just in reference about dissolvable microneedles. There are a number of ways. Again, it depends. I mean, you have to know the mechanical strength. Mm-hmm. You have to know the ability to co-formulate and coat it. You need biocompatibility, et cetera, et cetera, et cetera. So I'm answering more than what you've asked.
Right.
Ours are metal.
Okay. Are they pretty rugged over time? Like, could these survive storage for a year and more?
Yeah. So
you don't have to worry about the needles breaking.
There's two parts of it. So one is the microneedles themselves. If we make them ahead of time and we have the so-called ready-to-coat patches.
Right.
There is an equilibration that occurs just because of environmental things. And so what we're concerned about, because we do coat these microneedles, is looking at the surface energy of these microneedles. And our formulation takes into account the use of wetting agents so that when we coat, we get good uniform spreading, and we've published this, to show that this is important to get uniform dose loading. But anytime you make a material and then you store it, yeah, you have to expect that it's going to change, but then you want to know what that steady state is, because it won't continue to change forever.
Right. Thank you.
Yeah.
Hi, nice talk. Question up here, I see your talk was all about a daily patch, almost a fast release, but yet I see a weekly patch up on your slide here ready for Japan. Can you comment on how that works briefly?
Can I comment on how that works? Yeah, we were fully committed to doing a daily patch, and we still are for certain markets with the right partner. Our Japanese partner got approval of a once-weekly teriparatide or Forteo-like PTH 1 to 34, and had excellent fracture reduction data. We looked at what it would take to go from a daily to a weekly in terms of patch change, and for us it was, I wouldn't say plug and play, but it was a pretty easy way to go. So we are developing a once-weekly patch for the Japanese market, but we do have the capability, and we've already finished phase 1, and we're doing the pivotal study now, just starting the pivotal study, I should say, in Japan, so that, again, we're taking advantage of opportunity where the partner has a franchise that's being developed and a market that's being developed, and so we're going to go with the flow. Again, it's within our parameters, so we can do either one. Actually, when you look at differentiation, you say, well, okay, I go from a daily injection to a daily patch. Is that enough? And then I go from a daily injection to a weekly injection, and I go to a weekly patch. Maybe that's even more. So this is all evolving. It can't evolve indefinitely because we've got to get to market. And we have to do that the way our partner wants to commercialize and market.
The PTH story will be a very nice case study from now, because there is 3 companies working on different delivery. You have the Unigene, they just set up PTH, oral delivery PTH bioavailability of, let's be generous with them, 10% or something like that.
Oral,
right?
Oral.
I think, yeah.
Well,
Aegis.
But by the way, that collaboration stopped.
Stop? Oh, I don't know that. I just heard the press release. Well,
Phase.
when it stopped?
When did it stop?
Yes.
December.
Okay. Then Aegis, the intranasal delivery, they claim 40% bioavailability, 35 to 40%, and yours also 40%. No, I'm just saying, 10 years from now, when everybody will be successful, we'll see what the marketplace will support, the oral, intranasal, or the transdermal delivery, what the patient will accept. Yeah. And then the other one will be the cost.
Yeah, so let me answer the first one, because the 2 oral products for PTH have just recently failed. One was
Why?
They didn't meet the primary endpoint, which was increase in bone mineral density. So this is about PK. In my mind, It's about PK, and it's about not only the percent delivery, but the variability. And there's high variability with nasal, there's high variability with oral, from what I've seen. You may have different information.
No, the nasal seems to be 35 to 40%. I didn't see the hard data. Huh?
I'm talking about variability.
Yeah, all right, yeah. So comment on the cost. I mean, the intranasal will be cheaper, obviously, than yours.
If you can do it, yeah.
Right?
Yeah, there have been a couple failed attempts at nasal. There's now one that's in phase 1, which is Azalon, which has kind of picked up the gauntlet again.
Yes.
So we'll see how they do. I mean, there are a lot of people who have tried to do PTH. We think we're on the right track to be successful. There are a lot of dead bodies behind us.
Yeah. But
That's life.
Can you comment on the cost? How much that will cost with the batch and the
Well,
device?
So the cost. Well, there's cost of goods, and then there's selling price.
Yeah, the cost of good we know of PTA. I mean, there's nothing new. The cost of your device plus the cost of good, you have to make profit. So how much you want to sell it?
If 80% margin, how's that?
I agree. Yeah.
No, that's
80% margin, that's the normal.
that is our business plan.
Yes. So how much will be there?
We'll have an 80% margin.
I think there's another question here.
Yeah, sorry, I just wanted to make one correction on that. The Unigene oral PTH didn't fail. It's still an active program.
It's an active program, but I don't know that it's active with the original partner.
That's true.
So, see, I am not wrong there.
It was a very successful trial.
Okay.
What we believe now?
You
The
can
fellow
believe
from
the
Unigene,
Unigene
I
guy.
think, is the one you believe.
I only read what I read in the.
So on the press release, they said they're working with the PTA.
Okay.
Let's go review the press release.
Okay.
All
It's
right.
fine.
We can.
I just have to agree with what Peter was saying about variability. I mean, it is one of the key problems with nasal and oral enhanced delivery is variability around Cmax tends to be very high. The data that you showed looked great. Looks like your variability is very comparable to the sub-Q injection, which is a good thing. I did have one question. You said there were no adverse events and no immune response. Do you mean that it was no different from Forteo? Or
There's no immune response with Forteo. There was no immune response with our product.
Okay.
Does that mean we'll never see an immune response ever, ever, ever? No, nobody can make that claim.
Okay.
You keep watching that like you do for any product. It's always there.
Great.
But so far, so good.
Yes. Well, thank you, Peter.
Okay.
Let's thank Peter again.